right, everyone. All right. Well, I just want to say hi and glad you could join us. I think people are still joining. I'm Ellen Hahn. I'm the director of the University of Kentucky Center for Appalachian Research and Environmental Sciences. It looks like most people that I see on here are members and know me anyway and know what we're up to. I just wanted to, before I turn it over to Aaron Haynes to introduce our wonderful speaker for today, I just want to put in a plug and remind you all that this is Climate and Health Month at UK. And some of you were lucky to hear Dr. Robert Bullard last week at the Center for Clinical and Translational Science annual meeting. He was amazing. This is kind of our series, but then next week we have the Wyatt Symposium, and Kara will put that link in the chat so you can register to hear Aubrey Miller and to hear our amazing, Dr. Haynes' amazing group that she's pulled together. And then lastly, I just want to remind you that if you need services from UK Cares, please go to our website, our research support portal, and put in your request. And Kara will put that in the chat as well. So I'm going to turn it over to Deputy Director, Dr. Aaron Haynes, to introduce Kim. Thanks, Aaron.
Thank you, Ellen. And everyone, hello. Happy Wednesday in April. We're happy to have you. And we are thrilled to have Dr. Kim Cecil with us today to present her very exciting and I'm going to say groundbreaking research in neuroimaging related to environmental exposures. Dr. Cecil is on the forefront of this field, particularly with lead and other metals. Briefly, she received her bachelor's degree in chemistry and mathematics from Kentucky Wesleyan College. Then she went on to receive her master's and doctorate in chemistry from Vanderbilt. Then she was recruited to Cincinnati Children's Hospital, where she's now the director of basic and translational radiology research. So I've had the privilege of working with her on several imaging studies, and I think you'll be as excited as I am to see the impact, the actual visual impact of exposures on the pediatric brain. So thank you, Dr. Cecil, for joining us. We're so happy to have you.
Thank you, Erin. Thank you, everybody, for giving me some of your time today. So what I want to do is chat about how I incorporate neuroimaging into established environmental epidemiology cohorts. I come from a different perspective by being in radiology. And so I'll sort of share with that some of how that informs my kind of different worldview. Just a little more background about me. I was born in Louisville. I was raised in Owensboro, and until I went to grad school, I thought I'd always stay in Kentucky. So, and after grad school, I went to Philadelphia, and I couldn't wait to get back to be recruited in Cincinnati. And I lived in Northern Kentucky for several years until I married someone who was established in Ohio. So I'll talk a little bit about my career here and the last 20 years and how imaging and environmental health has become the focus of it. So let me see if I can advance. There we go. So the Children's Environmental Health Centers, which was how in Cincinnati I kind of got pulled together, they published this report, and it was just a nice little summary about how children are at high risk for chronic disease. And a lot of this is due from environmental exposures. So whether it's premature births from air pollution, respiratory infections from environmental conditions, high lead levels affecting neurodevelopment, and so on with autism, the contributions of environmental chemicals that are now thought to be responsible. And all of these exposures puts a tremendous burden financially on our society. And thus it's very appropriate for us to look at the prenatal influence of these exposures on health, prenatal health, and then ultimately childhood. So traditionally, from my radiology perspective, we're thinking about disease. We do comparisons of disease populations to typical populations. And even some of this idea of disease kind of has influenced environmental epidemiology. So you kind of think about diseases, the threshold, that portion of the Gaussian curve that is in the disease or disorder qualification. And in 2012, David Bellinger published a couple of papers I viewed this as changing our strategy about how we think about this and how we think about environmental chemicals contributions to the neurodevelopment of children. And I kind of call this Bellinger rationale, that even a modest impact that does not push a child's neurodevelopment into the range of clinical concern, that disease portion, it cannot be dismissed as benign, because if the exposure is prevalent, the total number of IQ points lost in the population as a whole might be large, and the reduction in the intellectual resources available to society substantial. So with this kind of in mind, while he's thinking about how he looked at this and examined estimated IQ point losses, the full-scale IQ point losses from either preterm birth, lead, organophosphate, pesticides, even down to methylmercury, you see that the environmental contributions are significant, and how they reduce IQ points across the population, not just the extremes of the curves. So with that in mind, my efforts and those of my group are to help reveal the contributions of neurotoxicant exposures to neurodevelopmental and mental health disorders in children and young adults using neuroimaging-based methods. So instead of IQ points, can I use neuroimaging metrics and quantify changes in the brain from these exposures? So since I work at a children's hospital, I can always do fun little graphics. So I've got a nice little neuroimaging toolbox. So while it's not quite this simple, I want to give you kind of a high-level overview of the neuroimaging toolbox that we use. And I start first with the toolbox that I developed my expertise in, and that's called standard MR spectroscopy. And this kind of comes from my chemistry days of using nuclear magnetic resonance, the NMR, to determine if I synthesize the proper chemical. But when you translate that into the human body, you basically see water and fat, but you can probe in between the water and the fat signals to see specific neurochemicals. And what we do is we sample either a single volume of interest called single voxel spectroscopy, or we can do techniques that sample the whole brain. But for simplicity, I want to just start with just kind of this simple cubic region. And we can see a variety of neurometabolites, one of which is N-acetylaspartate. And this chemical is found in neurons and axons that are viable. So if you have a healthy brain, healthy functioning neurons, that peak is at a certain level. And anything that damages the brain will reduce that peak. We see also kind of high-level neurotransmitters such as glutamate and GABA, but they're kind of combined. They have a similar methylene structure, and so they sort of fall in this one region. So it's a little harder to disentangle with this method. We see a peak from creatine and phosphocreatine, and so that's the cellular energy marker. So again, things that compromise that energy metabolism will have a reduction of creatine. Choline is derived from phosphocholine and glycerol phosphocholines. So all of our cell membranes have a choline constituent that I can measure there. Another peak we see is called myoinositol. This is a glial cell marker. So the glia are the supporting cells for the neurons. So anything that disturbs them, sorts of inflammation can change the myoinositol signature. And so with this kind of chemical fingerprint, I can get a profile for an individual's brain. And then with the epidemiology aspects, we combine them together, relate them to exposures, and understand metabolism of the brain. There's a more advanced tool called edited MR spectroscopy. And underneath that creatine peak, while here it's a nice little single peak, there's actually glutathione and GABA signatures underneath that resonance. So I play with the MRI scanner, do some pulse sequences, do some post-processing tricks, and kind of suppress the creatine and other metabolites to get at these novel chemical markers. And that's one of the things I'm doing in some of our current studies. So this is kind of cutting edge spectroscopy. And then probably what's the more exciting is the neuroimaging. And so if you go to your local hospital or an imaging center, they will do anatomical imaging. And that's just kind of static, looking at just structure of the brain. And what we've been able to do with our studies, and many others do this, is you take all those individuals' anatomy and then you harmonize them to combine them. You get rid of some of the differences that don't matter. You get rid of the skull. You get the brains into a comparable fashion. And then you can do group statistics, compare them to environmental exposures, and then other factors that it could influence: brain volume, cortical thickness, cortical surface area, so birth weight, the age of someone at the scan. Those factors can come into our epidemiological model, but yet we can then pull out information relevant to an exposure. So I'm just showing you some examples where we had reduced volume, and I'll talk about all of these in more detail within the context of the populations. This is to illustrate changes in cortical thickness. And let me just sort of show you kind of behind the hood in a really simplistic fashion. There's all sorts of scientists revising this kind of approach. This is something called statistical parameter mapping, where we do voxel-based morphometry. So instead of looking at the brain, when I first started doing this, we would draw regions of area of interest and just do manually trace the brain to get volumes. But that's not the best approach to do this. We make computers do this with different software programs. And just like when you take a photo, you've got pixels, your resolution. We have high resolution imaging, and we make the computer do comparisons of each of the millions of voxels within the brain. So we take our anatomical images. We generally have a few hundred slices within a human brain, depending on our resolution. We normalize that for some differences in the signal intensity. We segment the brain into gray, white, cerebral spinal fluid. We do a smoothing, so we're not just comparing individual ridges and gyri. There we go, some smoothing. Then for the hundreds of slices with the millions of pixels, we can get groups of individuals that we're studying within our cohorts, and then combine and compare with regression analyses at each pixel, or voxel, as in voxel-based morphometry, and do our studies. And that's kind of the basis for some of our volumetric cortical thickness, surface areas. And that's kind of just represented again here on this left side of the screen. So in addition to the segmentation, we can do a surface reconstruction of the brain, showing changes in volume. And so that's kind of, again, more of a static thing. You probably have heard about functional MRI. And again, without going into a bunch of physics and engineering, basically we are doing low resolution brain imaging, but we've got a high contrast, and we can do the images very fast, so we can do the time series. And what we're trying to do with functional imaging is when the brain is activated to do a task, whether it's to move your finger, to speak, there is a local change in blood oxygenation. So the brain says, I need oxygen to perform this task in this region. And when the brain extracts the oxygen from the blood, there is a local change in the magnetic properties that changes the signal in that region of the brain. And with fMRI, we're looking at that and that signal change and measuring that, assigning its geographic location in the brain. And that is kind of the basics of the bold oxygenation level dependent fMRI. And there's a lot of engineering to be able to do this, but there's kind of two branches of fMRI with an MRI based technique. and one is task-based and one is resting state. And a task-based can be something as asking someone to name verbs. So we present them with a noun such as ball or brush. And we ask people to think of verbs that what would you do with the ball? You would throw the ball, you would catch the ball, you would pick up the ball. When someone is doing that, they're activating the portion of the brain responsible for language. So that's kind of one mode, a specific task. You can do memory tasks, you can do visual checkerboard, so flashing, and so you would highlight the visual cortex of the brain. And then opposed to that is the resting state. And that's where you ask someone just to sit quietly in the magnet, kind of relax, just stare maybe at a plus sign or something. And what it is doing in that instance is you're not doing any sort of task, but you can measure. If you think of the brain as sort of like an idling vehicle, if you're at a stoplight, you have the potential to very fast take off, turn right, turn left, to go. And your brain actually has resting state networks that are ready to respond in milliseconds to a threat, to an action, to a decision. And here are just some examples. These are all just resting state networks that have that blood oxygen level just going at a low level. And you can take advantage of this and not be dependent on how well someone performs a task. And so this has opened up a whole new doors for studying children, infants, even sleeping infants under sedation. You can measure these networks. So again, I'm showing lots of fMRI because it is very complex. A lot of different people do different things depending on their expertise. So I just mentioned about the functional MRI. There's one more technique we've incorporated into our work. It's called diffusion tensor imaging. And that is making use of the fact that the white matter in your brain connects to the cortex where all the action's occurring, but those white matter highways are kind of like electrical wiring in your house or building. And it's got, you've got your copper wire and you've got insulation around it. So the white matter in your brain is similar to that. You have axons that are insulated, and that white matter is connecting all those regions. So different regions of the brain are interconnected, and you can measure this. Your corpus callosum is this kind of swirly thing here, and we'll go into a little more detail. But all of this kind of basic work has now become more complex into networks. And you can have structural connections in the brain that link disparate areas, and you can have functional. And so folks are now, this is kind of the leading edge, is doing different graph theory analyses to define these structural and functional networks, and then determine how the brain functions. And then we use that to study environmental exposure's effects on that functioning. And again, I'm just showing lots of pretty pictures. This is, again, is the diffusion, kind of showing the network of white matters. This is another, in addition to this statistical parameter mapping approach, there's something called free surfer, and that's determining volume, cortical thickness, cortical surface areas. This is actually showing kind of nodes within the cortex, and these are kind of these curves for measuring activation. And so you have the computer look for similarities and determine in which areas are functionally connected. And again, you can create these connectivity maps and connectomes and characterize, again, create more fingerprints to understand how an individual's brain is, and then in group analyses, how does it compare? So let me get into some of the work that we're doing in our cohorts. I actually started with when I was a postdoc at the University of Pennsylvania. My mentor came, and he had interacted with a woman who was seeing lead-exposed children in Baltimore. And so we brought in 16 of those little boys and girls and compared them to their cousins who were not as lead exposed. And we found reductions of N-acetyl aspartate, our neuronal marker in the frontal lobe. And this was kind of the basis of our work. I got simultaneously recruited to Cincinnati And your neighbor to the north here, we have a population just over 2 million, with about 12% living in poverty. We've had a long history. Let me back up. Let me change gears just a little bit. With Cincinnati Children's and the Interact for Health group in 2017, they did a child well-being survey and found that among about 5,000 people interviewed, about 13% of their children had asthma, 13% ADHD, 12% anxiety, 5% depression. So these are kind of some of the major morbidities of children that I was speaking of earlier that become chronic morbidities. And then when they interviewed these folks, the adults in this group, how many of those adults have these conditions? So asthma remains chronic, depression, and then with adults, you throw in some more high blood pressure and high cholesterol. And that's kind of a survey of disease in greater Cincinnati and northern Kentucky. And we know from being environmental scientists that these exposures are probably some of the basis for those chronic diseases. I like this little picture of Keep Cincinnati Beautiful, but we've got this tragic history of lead exposure in our city. I like this picture, even though it's a bit older. It just shows all those highways that are crisscrossing in downtown Cincinnati. And so it makes us ripe for these environmental cohort studies. 7 of every 8 housing units in Cincinnati proper were built before 1980. In our county, 7 of 10. We have air pollution from our multi-lane major interstates. And then thank you, P&G, for all the consumer products that you now have given us that have all kinds of environmental chemicals. So again, I have my toolbox that I'm applying to these cohorts. And I'm going to talk to you today about 3 of these cohorts. The Cincinnati Lead Study, where the mothers were recruited, the children were born between 1980 and 1985. We have the CCAAPS cohort, which is the asthma and air pollution study. Grace LeMasters started and Pat Ryan has continued. Births between 2001, 2003. This one is an infant cohort. The health outcomes and measures of the environment study. So Bruce Lamfield, Kim Dietrich started this study, a new birth cohort. So we have maternal exposures, as well as infants longitudinally followed. The kids were born between 2003 and 2006. We've now just started in the last 6 months a combined home and CCAAPS to look at air pollution. And then of course I work with Aaron for the CARES cohort. So let me give you a brief overview of the Cincinnati Lead Study. I have these pictures in here to remind me to tell you, one of the neat things about this study is that the folks, Kim Dietrich and his team at the University of Cincinnati, were in the community where they were recruiting. This Finley Street Clinic on one side of the building, UC had some of their primary care, and on the other side was the Cincinnati Lead Study office. So the participants, felt very at home to come walk to this building in their community, have their clinical care on one side and their research participation, and they've just been tremendous research participants. So Dr. Dietrich and his team recruited mothers in the late 70s, early 80s. The children were born. One of the treasures of this study is that every 3 months, from birth to 60 months, there was a blood lead measurement obtained. From 60 months to 78 months, every 6 months. Dr. Dietrich and his team provide a host of cognitive, behavioral, gait, balance testing. And then I came into the picture when between 2002 and 2005 to do the volumetric imaging, spectroscopy, and some of the fMRI. Dr. Haynes and I were able to secure funding and get diffusion tensor imaging. And this was right at the time that diffusion tensor imaging, some of these advanced techniques were coming online. And then we were able to do a follow-up study. So the participants were right around 19 to 21 at this point, and then 27 to their mid-30s at this point for 2 imaging. One of the things that's really, when I'm writing papers and still talking about the lead study, people forget that at the time of enrollment, up until roughly these kids were about 10 years old, they would have been deemed in the safe lead zone from the CDC levels. And then with today's standards, now everyone would be highly exposed and needing intervention. And so it's always important when we're doing these cohorts to think about, where we are in this progression and how important this work is. That it really about, getting into cohorts, developing cohorts to understand what are the, emerging threats. So we never have a lead situation again. And again, just part of that same context. So all of these kids were in this high lead, the prevalence of blood lead levels above 10 micrograms per deciliter, they would all have been, and their blood geometric mean of their blood lead concentrations were in very, what would be moderate to high levels today. Thank you. The Cincinnati Lead Study, we were able to bring in about 250 participants for the entire cohort, but we, with budget planning and MRI being expensive, we had estimated we'd get about 150. We overshot just a little bit. The mean blood lead concentration of those 23 measures from birth to 78 months is around 13.3 micrograms per deciliter. A range for the mean values from 4 to 38. And I just plotted out here the different measures for the group so that and showing you that the men and the women weren't that different in their blood lead exposures. It peaks as expected around 2 years of age, but it did remain fairly high. The cohort is mostly African-American and their full scale IQs are lower than the national standard. So with our study, we hypothesized that brain volume would lead to a decrease with increasing childhood lead exposure. There was some work done in occupational exposure showing this direction, so it wasn't that big a leap. Again, as I was saying, I originally proposed we would do some manual tracing. Unfortunately, this method had come out a couple of years before that allowed us to do kind of a hands-off, let the computer tell us without having any bias. And just to give you sort of a feel for the data, so this is one of our lower participants. This is one of our higher lead participants. And if you look here in the lateral ventricles, you see they are a bit more plump. And that's just kind of a peek to show you that there's probably volume loss. And the thing is, when you go get an anatomical MRI, someone would report you would have volume loss, but they'd have no idea if it was from lead exposure or some other insult. So we have to do these kinds of cohort group analyses. And when we do this, when we do all the normalization so we can compare these brains, what we found with the Cincinnati Lead Study was that there was significant gray matter loss, particularly in the medial frontal lobe here that extended into the anterior cingulate. And on these renderings, you can see there's some other areas in the cerebellum, some parietal lobe, some other high parietal. And then I'm just showing you within a voxel, a pixel within this region is what our curve our slope of our curve looks like here for plotting mean childhood blood lead level, again, adjusting for age at scan and birth weight after evaluating other covariates. One of the things I asked my postdoc, Chris Brubaker, to do is we've got that mean childhood blood lead level. What is driving that? Is it an early life? Is it a maximum? And he was able to do an analysis, again, taking adult imaging at age 19 to 21 and comparing it to historic blood lead levels. And what we found was that these later childhood blood lead levels were kind of driving our results. So these are probably the most heavily exposed and most heavily exposed over a period of time. And at childhood ages or at school ages, when you're going into school 5-6 years, when you're having that demand, high blood lead levels, and then that cognitive load, that's probably where we're seeing a lot of strong effects of the damage from lead. And roughly it's somewhere on the order of 1 to 5% of gray matter affected. So you sort of think, okay, maybe I've lost 1 to 5%. Typically, when we have clinical pathologies, neurodegenerative pathologies, that's on the order of 10%. So it sort of explains, why these people with high lead exposures, they can do normal functioning to some degree, but the cognitive demand, that 5% loss does have an effect that can be measured from our neuropsychology assessments. We did sex interactions as well as separate analyses, and we found that the men seem to be driving the result. And we sort of know from some of the neurobehavioral outcomes that men experience more deficits, as we know from genetic conditions, the vulnerability of the X chromosome, that there's some basis for this, but the women are not unaffected. And some of the parietal and high frontal lobe results are present there. Some of our subsequent studies, we find out that the women when we do diffusion imaging have more effects there. So we think there's some biology going on that's interfering with how lead harms the brain. We didn't report it in our first paper because we didn't model it correctly, but there is white matter loss adjacent to those gray matter regions. The neurometabolic assessment that was kind of the basis for some of this work from my work as a postdoc, we sampled 7 regions of the brain to look at primarily N-acetylaspartate levels in the brain to see if they are damaged. And I write this as for each 1 microgram per deciliter increase in mean childhood blood lead levels, we found reductions in these concentrations. But you've got to remember that our population is roughly 13 micrograms per deciliter. And everyone in the cohort at one point had a blood lead concentration above 10 micrograms per deciliter. They had at least one time point that they would be at this high level. And so our betas here become 0.5, which is again, very significant to lose those levels of these neurometabolite concentrations. So 0.5 reduction in N-acetylaspartate that tends to be somewhere along 8 to 10 millimolars in concentration. So we have decreased NAA in the basal ganglion cerebellum. We have decreased creatine, so affecting cellular energy metabolism. And much to my surprise was to see decreased choline in the cerebellum, the frontal white matter, and the parietal white matter. And this is going to be important because this is understanding how we understand some of our diffusion results. And we've got decreased GLX, which is a combination of glutamate and glutamine. At this time, we couldn't really resolve those in the white matter, and the vermis. So we've got some decreased neurotransmission markers as well. So here's our diffusion. It's not the nice little fiber tracking. So at the time when we did the study, diffusion was being used to evaluate stroke, and where you would see decreased blood flow, you would see reductions in fractional anisotropy, and you would see increases in mean diffusivity. So this is kind of how well the white matter bundles are packed. So we've got some form of injury here shown in blue. The mean diffusivity kind of gives you a magnitude of that injury. Axial diffusivity is going back to my analogy of electrical wiring. So the wiring, the white matter fibers themselves are damaged here in the centrum semiovale. And the radial diffusivity is our marker of the insulation on, it's the fatty choline bearing insulation on white matter, the myelin. And so while this pattern is very expected for injury, and here this red and yellow is very expected from what I've known from my clinical side, the blue was very unexpected. And when we look here at a lower level in the brain, this is at the level of the corpus callosum, we have this increased myelination. So what does this mean? Excuse me. Increased myelination. Well, the way we've interpreted this with our diffusion results is that with our fMRI results, let me jump ahead a little bit. So we asked the participants in the Cincinnati Lead Study to do a verb generation test. But as I mentioned before, think of action words when we present nouns to you. And we have the typical areas for where language function is, Broca's areas and Wernicke's areas. And this is reversed. This is radiological convention. So this is actually the left side of the brain. So this looks pretty good. However, when you probe within these ROIs, you see with increasing blood lead levels, reduced activation in those regions. But to our surprise, there's these areas on the contralateral hemisphere that have increased activation. And when you kind of map the circuit, if we go back to our diffusion, that circuit goes through the splenium of the corpus callosum. So what we have is some attempt to recruit the opposite hemisphere to perform the language task and the functioning. So this was very interesting that while the brain is suffering insults from lead, it is also trying to adapt to compensate. Some other work that was done with the Cincinnati Lead Study throughout adolescence, early adulthood, and later adulthood by my colleagues Kim Dietrich and John Paul Wright was to understand criminality and the risk of childhood lead exposure to criminality. And in the last version of their analyses, taking the whole life course from 18 to 33 years, they found that the childhood blood lead concentration prospectively predicted variation in adult arrest over the life course. And on the same sort of theme, while we didn't really appreciate from the imaging the prenatal blood lead driving this result for the arrest and the effects of lead, it was these later childhood lead. So late childhood, excuse me. Late childhood, meaning from 60 months, 5 years through 60 months to 78 months. So 6 and 7 years driving the results. So sort of like what I was seeing when we did our comparisons of volume in those later childhood blood lead concentrations are really the ones kind of driving a lot of the imaging findings and some of the criminality findings. We've tried to kind of wrap the story up. So with the psychopathy measures and the criminal arrest analyses, linking them to our imaging. And just to kind of summarize it, so the second session, finding gray matter regions where there's volume loss, the association between arrest and volume is much more striking. And then Travis Beckwith did this conjunction analysis to show that this frontal lobe, the medial frontal gyrus, the anterior cingulate gyrus is driving some of this results. These injury to the brain is involved in this aspect of criminality. So the improper functioning of the brain. And how this manifests is a little more complex with the criminal justice, the psychology, but there is indeed some level of brain involvement with this. Just a kind of a side note, the lead story is continuing. And then there was this recent report from JAMA Pediatrics where I believe it was Quest did an analysis of their data of over 1 million children from their capillary blood lead testing and looking at the percentage of children with detectable blood lead levels. So Kentucky's got a value of 70 here, Ohio 52, and a percent of children that were tested in their laboratories, and percentage of children with elevated blood lead levels. So Ohio is higher than Kentucky at 5.2. Kentucky, I believe this is 1.6. Sorry for the small size there to show this analysis. And it just, the results of this study was showing that the odds of detectable elevated blood lead levels were significantly higher with those living in pre-1950s housing and in poverty. And when I saw this, it reminded me of one of the basis study, or one of the original Cincinnati Lead studies, showing that the type of housing that the participants of the Cincinnati Lead study was driving the results of their blood lead concentrations. So that deteriorated pre-World War II housing is responsible for this lead exposure from decaying paint. So it's still a problem where our children live. So let me switch gears, and in the last 20 minutes here. So traffic-related air pollution is strong. We know it's strongly associated with adverse cardiopulmonary health effects, but what is it doing to the brain? And this is still a growing, emerging field. When we first wrote our grants for studying this, we were hypothesizing, is there translocation from the respiratory system? Is there direct input into the brain from the olfactory nerve, or is this some sort of neuroinflammatory mechanism, or is it some combination of all three? So we've been working with the CCAAPS cohort. Sorry about this. Our lovely Cincinnati allergy season here kicking in. So this was the cohort Grace LeMasters, Pat Ryan, our PIs of. So children were identified from public records. They were recruited based on whether they lived close or far away from a major highway. There's some allergy and asthma. Allergy and asthma were the driving force for this study originally, but there's a lot of rich data, including determining the elemental carbon attributable to traffic. So this is more diesel driven types of air pollution was the focus of their analyses. And then at age 12, about half the cohort came in to be assessed for neurodevelopment, as well as a nested study to have MRI comparing the highest and the lowest quartiles. So again, simple hypothesis, we tested that TRAP would be significantly associated with changes in brain structure and organization, and there would be critical windows. We had about almost 150 participants at age 12, and just over half were considered high exposure. So analogously to the Cincinnati Lead study, we did high resolution anatomical T1 weighted imaging, and the classification is the TRAP, the elemental carbon attributable to traffic exposure at time of birth. And what you'll see here, there is a lot of cerebellar reductions. This is on the order of about 3% of the brain gray matter volume is reduced. And there's also some of this pre-central, post-central gyrus, some higher frontal lobe volume loss, but not quite as striking as what we saw with the lead study. We saw about a 4% reduction in cortical thickness with our measurements. Again, the precuneus, the motor regions, the premotor, the somatosensory motor are the regions affected. We performed spectroscopy in this, and one of the things we were looking at is not just understanding how has the brain just changed, what's the functional component of this. And we found from our analyses, and I know Pat Ryan came and spoke with you, that measures of anxiety and depression were elevated in this cohort. And we wanted to see if we could kind of link some of this. And the myoinositol concentration, as I mentioned before, is a glial cell and inflammatory marker. And with increased myoinositol in the perigenual anterior cingulate frontal lobe location, this correlated very nicely, increased myoinositol with increased anxiety scores. And Kelly Bruntz published this on our behalf, and I know Pat went through this as well, linking the ECAT exposure to age 12 to myoinositol levels at age 12 and generalized anxiety symptoms at age 12. So lots of 12s there. But I emphasize this where the structural findings seem to be driven from early life. The metabolism is It has a structural component. So if structures in the brain, if you don't have neurons in the brain in a certain region, you're going to have a low NAA. But spectroscopy also gives you a snapshot of some of the current metabolism too. So it can be a little more complicated in interpreting, but this was a nice support of this. There's an inflammatory aspect to air pollution. The cohort that I'm doing a lot of work with now is the home study, and we're still in the early stages, but I want to just give you kind of coming attractions kind of approach. So the home study was very nicely designed, and it gives us a rich database of primary exposures, flame retardants, old flame retardants, the polybrominated, and the new, the OPEs. We have polyfluorinated alkyl substances. We've got heavy metals. We have air pollutants, pesticides, and phthalates among our exposures. We have a number of time points: the prenatal 16, 26 weeks, birth, toddler 1 through 3, childhood 4 to 5, mid-childhood 7 to 9, and pre-adolescent at age 12, which is nice because the CCAAPS and the home can be combined together because we did very similar protocols at age 12. Our key outcomes are anxiety, depression, some social skills, and executive function. And let me just give you a little more recap. So, and a nice, really strong study team of study coordinators, and Kim Yolton leading the efforts. Nice little chart here. We were doing our progress before we finished. We ended up, out of 468, we ended up with 256 completing all the measures at phase 4. But phase 1 was just the development of the cohort, assessing infant and early child neurobehavior, health and growth, continuing that through phase 2. Again, the emphasis is more externalizing behaviors. Lead was kind of the chief exposure. And then we have switched more to a flame retardant and other chemicals. Phase 3, Iman Chen was able to secure funding for flame retardants. And then at phase 4, Kim Yolton and I shift this focus to more internalizing symptoms and adding neuroimaging. But also Joe Braun has been looking at adiposity. So it's really nice to have multiple grants funding the same visit. It made for a very long day for the participants. It was about 7 to 8 hour study visit at age 12, but you can see we were very successful in getting a large component of the cohort to come in and give us stool samples and teeth and do blood draws. And we ended up with 199 completing their MRI. I just stole from the American Chemistry Council, not stole, I'm giving them credit, just some little surveys. So it's a very positive about flame retardants, but it's just very concerning that since 1980, the change in all of the chemistry to do these flame retardants that don't have a covalent bond, they just are on the surface of so many things in our homes and our lives. Throughout our homes, our workplaces, we're just bombarded with this. And I've got some preliminary data to show you. This has been submitted. Jeff Strawn, a psychiatrist working with us, who I've worked with for several years with his clinical populations with anxiety disorders. We've done some analyses relating the PBD exposures, and you can see most of our families and children are above the limits of detection. And what he's seeing in his analyses, he and Ying Wu, an increase in SCARED, the measure of anxiety, the screening for anxiety-related disorder scores. There's the increase, statistically significant increase with doubling of PBD exposures. There's a risk of having clinical threshold above 25 of clinical significance associated with doubling the PBD exposures. And then even an increase in the depression scores, though not quite as striking as the anxiety. So my first analysis is trying to look at what's the relationship with the brain in these this exposure and anxiety outcomes. And my former postdoc, David Edmondson, he's now junior faculty. This is his work where he made this nice little figure illustrating what we have done with our free surfer volumetric and cortical thickness analyses. So for measures of anxiety, we have changes to the thalamus kind of illustrated here in this cartoon. David has done a nice job putting it back into my hands because a lot of this is just computer animated. And I'm just sort of showing you some of our preliminary data. We're working out what's the best metric for some presenting the PBDEs. So there's some different patterns between PBDE 153 compared to the other metabolites or congeners, excuse me. So we see a relationship of the thalamus volume with PBDEs. We see a relationship of the scared anxiety outcome with the thalamus volume. So smaller thalamus volume with higher scared scores. We do some preliminary mediation analyses. This doesn't have any of the covariates in this. So we're building those models. Jane Curry and Mekabib Alte are working on the DAGs right now to complete these analyses, but we found a lot of subcortical changes there. And then these are the regions where we're finding cortical changes. So again, back in this pre-central gyrus, the post-central gyrus, there's something going on. There's something going on with the superior parietal cortex back here. So these are pre-motor or motor areas, post-motor areas, and there's some other anxiety and social behaviors. A postdoc, Kim Hartley, has published some of this relationship of gestational PBDEs with social skills and problems behaviors. And so far, all of our imaging analyses are just linking the gestational as opposed to some of the postnatal exposures. But back to Kim's analysis with caregiver reported social skills and problem behavior scales, especially for males, she found significance. And what we're seeing with this as well is when with the social skills scores, this superior parietal region, again, is really important, as well as the hippocampus. So higher scores for social skills have higher volume. Higher scores for problem behaviors have lower volumes. And the parent and the child scales are somewhat similar with our changes in volumes in the child. So that was good news. And again, we're finding this superior parietal region on the cortical parcellations related to these social behaviors and PBDE exposures. So whether this is a link to understanding autism or anxiety disorders, we don't know yet, but we're hoping we can answer that. So let me summarize here and thank my collaborators. They've just been tremendous. They've been the best people to work with in this study. So let me stop there.
Awesome. Thank you, Kim. That's absolutely fantastic. So please do ask questions in the chat. I'm seeing a few come in. So Kim, we'll just start right away. We have a question from one of our clinical researchers. Can you talk about the blood-brain barrier and how it serves to protect the brain? Does lead go through? When is it formed? If you could talk about the blood-brain barrier.
I'm not an expert at all on this, but all of these exposures are getting in there, and it almost is, how much is placenta protection? As I know, in utero, it's very underdeveloped and it's very leaky. I don't know the answer to that specifically for lead, because our results were not showing. We saw more postnatal lead influencing brain volume. So I'm sorry, I'm not great on that.
Well, to your finding of age 5, 6 lead level influencing imaging, do you think would you say that's do you think that's reflective of the actual concurrent measure itself, or a summary of the past? Because I know those children, had very high levels all the way up to that point, but that's kind of like a, the total weight of the lead in the brain.
Yeah, the way I think about this, so you have all the development in utero, you've got your glia cells in utero migrate out and they form the cortex. But the brain from birth, there's still things going on, still developmental changes, but by about 3 years, everything's kind of intact except for the myelin. So all that white matter is kind of unprotected to some degree. There's certain structures, this corpus callosum is myelinated relatively early, but the main cerebrum, it takes until your 20s to be myelinated. So all these Cincinnati Lead Study kids have again, that graph showing that through age 6.5, they have high blood, they're in the chemical soup of lead is still very high. So what I think happens is what we're doing is, and looking at them instead of concurrently looking back, the structure has been altered and changed, and we're probably seeing the tip of the iceberg. It's probably there at age 2. We're just not sensitive to detect it because the change has already happened. And with our looking back while studying them as adults, it's kind of I'm sorry, I'm struggling for the words to say, the association we're seeing is just reflective of the damage, but it's not really taken a direct snapshot. I'm sorry, I'm not conveying, I have an image in my head and I'm trying to describe it, but
I hear you working it out.
Things are happening at earlier stages that just have a residual effect that just continue on, that we can then later image and see. And it'll be interesting with like the CCAAPS and home. So we scanned both of those cohorts at age 12. We're scanning them now at age 17 to 21. We're going to do longitudinal analyses to see, are these same patterns persisting, whether it's behavioral patterns or imaging patterns. So if we showed that there's a reduction in cortical thickness due to air pollution at age 2 or at age 12, that's 2%, is it increasing, or is it, is it the same, or is it increasing? And sort of, we might be able to tease apart prenatal effects from postnatal effects.
I think it's amazing. There's been so many neurotoxicity researchers focused on IQ and outcomes, and you've been able to capture that into the actual effect on the brain itself, not just the measure outcome of it, but the actual structure, which is
the structural basis for all these behaviors, or even IQ.
Yeah,
it puts aside a lot of the other factors and kind of links it back that if the brain isn't formed correctly, or is damaged postnatally, it's not going to perform correctly. So.
I know we just had our keynote, Robert Bullard, come in for the CCTS, and I can, there's definitely environmental justice issues going on here, especially with the lead study cohort. But Living on Earth did a very nice, wonderful special on your study with Dietrich study with the crime and criminality. So I found it and I'm putting it in the chat if people want to watch that episode. So Ellen asks a question on neuroimaging related and radon. Have you ever looked at radon exposure?
No, I haven't.
And then
is there a cohort somewhere where that's really been documented?
Yes, Ellen has, Kentucky has known levels of radon that are concerning, and she has building a cohort. So probably you will discuss.
Yeah, I can talk to you about that. Thanks, Kim.
Cool. Very neat.
One last question, because I know we're at time. Jason Unrein would like to know, what about report back? Do you report back the imaging? How do you do that?
Well, with the Cincinnati Lead Study, we really didn't do that. With home, we haven't done it other than we review all the images for incidental findings of clinical significance. So all of those do go back to participants if we find something that has potential clinical relevance. I guess we probably have been unknown and we don't want to alarm people. So finding that right balance of saying, here's our group. And this is where it gets so tricky. And that's where I was trying to build this. People from around the world call me and say, my son's in prison. I know he was exposed to lead. Can you come help? In his legal thing. And it's individual versus group analysis. So I'm struggling with that. But it's something as we, I guess, grow more confident and more people replicating, we probably will need to do a better job of that.
But when people are in the scanner and if they do see something clinically relevant, they are notified.
Yes,
Correct.
yes, definitely. Yeah. Yeah. And
Right.
and sometimes it's kind of like that breast cancer, mammography thing of
Yes.
we don't know what it means. And it just causes a lot of alarm. And again, the Cincinnati Lead Study, knowing those folks didn't, and at the time there was no Affordable Care Act, there was most of them didn't have insurance. So when I had to call and say, we think that the cerebellum is dipping down too far in your neck, and that might be harmful. We had to work with them, and fortunately, we had folks at the University of Cincinnati College of Medicine who would see them for free.
That's
and
awesome.
evaluate them. For things that they were probably fortunately, most of them were born with all the incidental findings.
Yeah.
And it was just to notify them, but it causes a lot of angst, especially when we're the 12-year-olds. We were seeing like that condition where the cerebellum goes down into the neck just a little too far, a millimeter or two. It's called a Chiari, and it causes a lot of angst when you say you need to consult with a neurosurgeon. And but Chiaris are something, arachnoid cysts are things that a lot of the population have, and until we started doing all this imaging of everyone, we didn't really appreciate their common incidental findings, most of the time of uncertain significance, but we do inform them and work with them in their situation.
Well, Kim, your research is phenomenal. We thank you so much for what you're doing to help kids that are exposed to chemicals unjustly. So thank you. And I'm gonna applause. Fantastic talk. Thank you.
Thank you. Thanks for bearing with me on my allergy here.
No problem.
Thank you, Kim. We appreciate you so much. That was really interesting. And I have no idea if that was a crazy question about radon exposure. There's so much we don't know about radon. And so, yeah. Anyway, I'll contact you another time. I have to run, but thanks again.
Very good.
Thanks, everyone.
Thank you.